afternoon. Good afternoon.
Good afternoon, sir.
Good afternoon, Sir Ordonio. Long time no see.
Good afternoon po. Sir Carlo po ba to?
Yes po.
Ay, hello, Sir Carlo. Sorry, nakabingan po yung screen ko. Actually, may sakit po ko ngayon. Balat ako at saka inuubo. So, it's fortunate na nakapagsalita ako ngayon, pero kanina, kagabi, hindi ako makapagsalita dahil tuloy-tuloy yung ubo ko. Makati. So, sana matapos mo ngayon, walang glitch.
Thank you po, Sir Ray, sa pagpa-conduct ko sa amin.
Oo naman. Nauuhan ko na eh, kaya tuloy lang.
Good morning, Sir Chan.
Good morning. Can you hear me all right? Hello.
Hello, John.
Yes.
I can hear you.
Yes, I can hear you. Sound loud and clear.
Let me put up the volume a little bit. Is this better?
Yeah. Sounds good.
Let's fix the camera as well. Here we go.
John, that's in your house, right?
Yeah, I'm still at home. It's 8 a.m. I better put up the background that you have. Looks good. Give that a try. Oh, here we go, Carlo.
Oh, there.
You'll see screen sharing works.
Enrique, is my background correct or should I turn on mirroring?
Yes, Sir Carlo. I think it's fine.
Okay. Thank you.
Hi, 30. 30. Hi, Sir Carlo.
Yes, sir.
Sir Carlo, can we start at 2:15?
I think Raymond's concern is that we finish at yeah,
We're
yeah,
still
yeah.
waiting for the
We start
participants to
at
come in.
we start at 2:15 and aim to finish at 4:30.
Okay. Yes.
Yeah, because as Raymond pointed out, if we take too long, people will start worrying about catching rides in their office shuttles, and basically how to go home and battle EDSA traffic. So, yeah, we could start 2:15 and then aim to finish at 4:30.
Okay. Thank you.
Magandang hapon from Violi, Villegas.
Good afternoon po, Ma'am Bion.
Hello, ma'am.
Hello.
Long time, Violi.
Who was that?
John Coleman here from the
Hi,
Netherlands.
John.
Long time.
Yep. You look younger than the last time I saw you.
I guess I need a new camera then.
I thought it's the haircut.
Thank you.
Welcome everyone in this webinar entitled Philippine Biosafety Regulations on Gene Editing Technology, organized by the National Committee on Biosafety of the Philippines, or NCBP, and the Program for Biosafety Systems, or PBS. 2 weeks ago, we conducted the first part of this webinar series that was focused on the new technological advancements, burgeoning in the applications of new breeding techniques or plant breeding innovations. Today, we will have the second part of this webinar series that will highlight the biosafety regulations for the use of gene editing technology here in the Philippines and abroad. So the objective of today's webinar is to first provide an update on the current status of research and development on gene editing in the country and at the global scale, and present the local and international regulatory framework for the use of gene editing. So we will learn more about these topics through today's talk from our esteemed resource speakers. So good day, everyone. My name is Enrique, an SNP Fellow of NCBP, and I will be your moderator for today's webinar. So we are live at here at the Department of Science and Technology Central Office, and we are also being simultaneously being broadcast via the NCBP YouTube page, and of course via Zoom. So thank you everyone for joining us today. So before we start, let us first have the national anthem. So to officially start the program, welcome message will be delivered by Ms. Maria Lorelai Albagala, Assistant Scientist of the Department of Science and Technology and Head Secretariat of NCBP and DOST Biosafety Committee.
To our distinguished guest resource persons, ladies and gentlemen, a pleasant good afternoon. We are delighted to welcome you to the second installment of our webinar series titled Biosafety Regulations on Gene Editing Technology, organized by the Department of Science and Technology, National Committee on Biosafety of the Philippines, in collaboration with the Program for Biosafety Systems. Our mission with this series of webinars is to advance the cause of biosafety in our country, and today's session is a significant step towards achieving that goal. We have a compelling agenda lined up for you, designed to provide valuable insights and updates on the latest developments in gene editing technology and the regulatory framework surrounding new breeding techniques or plant breeding innovations. As we navigate the landscape of biotechnology, it is crucial to stay informed and aligned with international standards. This webinar serves as a platform to explore the current status of research and development in gene editing, while also shedding light on the regulatory guidelines and global trends in the use of new breeding techniques or plant breeding innovations. The knowledge and expertise shared here today will undoubtedly contribute to our collective understanding of these critical subjects. Once again, welcome to the Biosafety Regulations on Gene Editing Technology webinar, and let's make the most of this opportunity to learn, collaborate, and contribute to the advancement of biosafety in the Philippines and beyond. So without further ado, I encourage you to sit back, relax, and prepare to dive into the world of biosafety regulations and gene editing technology. We have a panel of esteemed speakers who will guide us through this enlightening journey. Thank you for joining us today.
Thank you, Mamadou, for that warm welcome. So before we welcome our first research speaker, please note that we will be holding an open forum after the presentation of each speaker in order to address all your questions that you may have. So you may submit your questions in the Zoom chat box or in the comment section of the NCBP YouTube page, or you can just raise your virtual hand during the open forum. And also, do not forget to fill out the attendance sheet posted right now in the chat box. Okay, so let us now proceed to the main event, which is the presentation of our esteemed resource speakers. So it's with great pleasure to introduce Mr. Jan Komen. Mr. Komen is the Assistant Director of PBS, Program for Biosafety Systems. He works as an independent advisor to research and regulatory organizations around the world on matters of biotechnology policy, biosafety regulation, and research program management. His areas of expertise include biosafety policies and procedures, intellectual property rights and IP management, international collaboration and technological transfer in biotechnology, designing, training events, materials, and methods for policymakers and research managers. Since the launch of the USAID-supported program for biosafety systems in 2003, Jan serves as PBS Africa Coordinator, with specific responsibility for supporting the planning and implementation of technical assistance activities with partner countries in Africa. Mr. Komen's work as policy advisor for agricultural biotechnology and biosafety is instrumental for his presentation today. He will talk about the global developments in genome editing, applications and regulations, implications for emerging economies. Everyone, let us all welcome Mr. Jan Komen.
Thank you so much, Enrique, for this excellent introduction. No need for me to add anything here. Let me share my screen. And I suppose it's going to work. I hope you can see it okay.
Yes, that's right. Thank you.
Excellent, excellent. So in the next 20 minutes or so, I will be scratching the surface of global developments in genome editing applications and regulations. And we'll point to some implications for emerging economies, as we call. My name is John Komen, and I'm the Assistant Director / Africa Coordinator for the Program for Biosafety Systems. As you all know by now, much of my work these days is focused on Sub-Saharan Africa. But in the early days of the program, I was a regular visitor to the Philippines, and I'm still impressed. I was always impressed and still am about the leadership that the Philippines have expressed in the area of agricultural biotechnology, biosafety policy development, and now more recently in genome editing. And I think I can stick to a general statement here, but I think it's also important to point to the leadership that key individuals have always shown in the Philippines. Some are with us today. I saw Dr. Villegas, I saw Dr. Ramirez. Dr. Ray Ebora is one of the founding fathers of our program and was instrumental in launching the program some 20 years ago in the Philippines. And it's been an adventure and a great pleasure of working with institutions and individuals in the Philippines since then. But let me start with my summary of the presentation. I will give you a brief introduction of our program and what the main points would be that I would like to cover in my presentation. I will be pointing to the very rapidly evolving context in research and development, as well as in policy development regarding genome editing. Towards the end of my presentation, I will be presenting some policy considerations for research directors, scientists, and most importantly, for regulators. And before that, I will be showing a case study from the European Union that aims to illustrate the importance of taking a product-based approach versus a process-based approach. And then we'll wrap up also in the discussion with some emerging best practices. If my slides are not transitioning or if my voice is not clear, please interrupt me anytime, huh? But I suppose it's okay for now. Good. So 1 minute on the Program for Biosafety Systems, or PBS. PBS started in 2003. We recently, together with Carlo Custodio, who's in the room today, and who is our team leader in the Philippines, we celebrated 20 years of PBS. It's almost unbelievable. And PBS is USAID supported. It's managed by the International Food Policy Research Institute in Washington, D.C., which is one of the sister centers of IRI, the International Rice Research Institute in Los Baños, and a member of the One CGIAR consortium of International Agricultural Research Centers. But as you all know, IFPRI focuses on agricultural policy and agricultural policy development, which is why PBS has found a home there. So over the past 20 years, we have been providing technical expertise and guidance, hands-on training on biosafety, food and feed safety topics, legal analysis and policy support as guidelines, regulations, and laws are being developed. Very importantly, regarding genetically modified organisms and biosafety aspects, we have supported organizations as they communicate about their decisions, about biotech, about biosafety, based on a solid stakeholder analysis. Of course, in all of our work, we are able right now to build on a safety track record of more than 25 years. Since the launch of the first genetically modified plant variety in the United States, well over 25 years ago by now, a lot of information and expertise has been gained and experience with the crops at hand. And I think we can safely say that millions of consumers have been exposed to genetically modified organisms now. Farmers have planted millions of hectares. We know pretty well how these organisms behave in the field and what impact they have on human health and animals. So this is PBS in a nutshell. The program continues to evolve, which is why genome editing and associated technologies are more and more taking the spotlight in our program more recently. So turning to the topic of today, genome editing, what I would like to emphasize, without going too much into detail regarding the set of technologies, the additions to the toolbox that we have. I think the webinar of 2 weeks ago did a much better job there. But what I would like to emphasize is that this is an extension of the plant breeders toolbox. And it's an extension of a toolbox that becomes ever more precise, from crossbreeding, plant breeding, which has been practiced for centuries now, to mutation breeding, such as chemical mutagenesis or irradiation breeding, which is quite a crude way to develop mutant crops with desirable traits, to the biotech and transgenic revolution that we witnessed in the 1980s and 1990s, to a very precise way of modifying a plant even without inserting a foreign DNA or without resulting in a novel combination of genetic material. And this is the key factor that we're going to discuss today as well. But important to emphasize here that it's a logical extension of the breeder's toolbox, also based on a foundation that we have with classical mutagenesis, with classical plant breeding, as well as with genetic modification. And an important difference with the GM revolution, as some call it, is that this is already resulting in a large number of actual products that are leaving the research and development pipeline and are entering consumer markets. You may be aware of some important recent product launches, such as the one by Pairwise and Conscious Food, where mustard greens have been developed that have a better taste. You may have heard of the approval of GABA tomatoes, healthier tomatoes that are being approved in and consumed, sold and consumed in Japan as we speak, or the high oleic soybean oil put on the market by Calyx. And it's important, and these are three illustrative examples of the types of products that are already entering the market that are gene edited and that are put on the market not as GMOs, but as conventional products that have been exempt from a full biosafety review in the countries where they have been approved. So if we compare that to the still limited number of crops that dominate the market for GMOs, soybean, maize, cotton, you're all aware of that. I think the developments that we witness right now in genome editing are much more diverse and also occurring much more rapidly compared to past developments in genetic transformation or in mutagenesis. And this is not just a handful of small companies who are active here. The international centers that are part of the One CGIAR organization, including IRI in the Philippines, the International Rice Research Institute, are quickly adopting genome editing in their research programs, already resulting in lab research as well as field trials for really important developments in key staple food crops. So taking rice as an example, IRI and partners are very much involved in genome editing, looking at enhanced disease resistance in rice, better food safety, safer rice with low arsenic or cadmium residues, nitrogen fixing rice, as well as better resistance to, for example, the brown plant hopper, which is an important pest in rice. And again, to show that the pipeline, the genome editing pipeline that we witness, not just in the private sector, but also in the international public sector, and I will also show later in the local public sectors, is much more diverse and much more innovative compared to the genetic modification era that we have witnessed. It's also important to emphasize here that this is not just restricted to the plant kingdom. Genome editing is also being applied in many research programs for animals, leading to to more robust species of chicken, of dairy cattle. We have already seen approvals for genome-edited fish on the Japanese market, tiger puffer and red sea bream, with improved growth characteristics have been approved on the market in Japan. But we also see important developments in salmon, in swine and goat, as well as other species in the animal kingdom. So if we again compare this to the green revolution era or the biotech revolution era, in genome editing, we see much higher diversity of species involved, including animals, including microorganisms, and so on and so forth. Now, if you're interested in knowing more about the research and development pipeline, there's a very important online database that monitors projects that involve genome editing worldwide. It's maintained by a European organization, and it's called e-Usage. And this is part of the network of European sustainable agriculture through genome editing. There's around 600 projects listed in this database right now that are beyond the proof of concept. And if you visit this database, you will find an excellent overview of the various applications of genome editing at the moment. And again, to emphasize it, it will confirm the wide diversity of actors involved, international, private sector, public sector, and so on, and a very wide range of species this work involves, not just plants, but also animals and microorganisms. But today, we're going to focus on policy developments and regulatory guidance. And this is where it really becomes tricky, because if we look at the available international guidance regarding genome editing, I should say it's very limited. Of course, many countries who are signatories to the Cartagena Protocol on Biosafety, which is a protocol to the Convention on Biological Diversity, take the protocol as the starting point. And the definition included in the protocol is therefore an important starting point as we consider regulatory approaches to genome editing. Because the protocol defines an LMO or a GMO as a living organism that possesses a novel combination of genetic material derived through the use of modern biotechnology. And those are the two key criteria that define a GMO. And this is really the key question that any regulator that has to consider genome editing applications has to ask himself or herself. You know, is a gene-edited organism possessing this novel combination of genetic material, and has it been developed through the use or application of modern biotechnology, which is also very specifically defined in the Cartagena Protocol on Biosafety. Now, suffice it to say at this point that the topic of genome editing and synthetic biology and gene drives and so on and so forth is hotly debated in meetings regarding the Cartagena Protocol these days. And to date, there's no clear guidance coming from this international forum and international body. So let's have a look at what some of the important emerging economies have come up with, which is an important yardstick for other developing and emerging economies to look at. And I think the Philippines, in determining its initial approach, has done that as well. And I think the most important and most significant developments here are actually taking place on the South American continent. And I've listed Brazil and Argentina here as two important countries that have a rich track record in genetic modification and the production and export of GMOs, such as soybean, maize and cotton. And especially Argentina was an early adopter of genome editing in its research programs, and also an early adopter of regulatory approaches to gene-edited organisms. And I would like to focus for a few minutes on the case of Argentina, since it illustrates the regulatory challenges and solutions very well. Now, this is the system that Argentina concluded with. In 2015, in a guideline on genome editing, Regulation 173 of the year 2015, they adopted a consultative approach and a decision-making checklist, decision-making tree that basically took the question of, well, does this application or this product possess a new combination of genetic material as the starting point? Well, if it's the case, if there's a new combination of genetic material, which is very well possible when you're applying gene editing or specific tools in the genome editing toolbox, if the end product possesses a novel combination of genetic material, the applicant is notified within a short period of time that the product falls within the scope of the GMO legislation in the country. If it doesn't, if it only uses a transgene temporarily, or if the final product is free of transgenes, then the termination could well lead to a non-GM status, which means the product is exempt from full biosafety review in Argentina. But still, although it doesn't fall under GMO regulations, it is still regulated under other existing environmental and agricultural laws. So the statement that is often made that we're now opening the doorway to unregulated biotech products by exempting them from GMO regulation is not quite the case. Even if a product is exempted from biosafety regulation and full biosafety assessment, it will still have to go through, for example, variety testing, seed certification, and so on and so forth. It just means it will be regulated elsewhere, but not as a GMO. The slides that I'm using, including this one, are actually provided by Dr. Martin Lima from Argentina, who's worked as a regulator in Argentina for many decades and now is at the University of Quilmes. So before arriving at this decision-making tree, Argentina spent a decade or so assessing its approach to genome editing, starting with a debate at the National Commission for Biosafety, leading to this first ad hoc regulation, which was the basis for the first assessments and decisions. And by 2021, the regulators had looked at over 20 cases of involving genome editing. And many countries in Latin America have followed this example, by the way. Again, it's very much based on the definition of an LMO under the Cartagena Protocol. It uses a case-by-case determination mechanism whether a product constitutes a GMO or not. And the assessment and the communication to the applicant is done within a period of 60 days. And as a result of this practical approach, if we compare the developer profile for GMOs with the developer profile of gene-edited products or NBT products, new breeding technology products, we see a shift from foreign applicants, multinational applicants, to local applicants, the local private sector, as well as local academia and the local public sector in Argentina are now much more active in developing products that involve genome editing, because there is a clear science-based and straightforward process to determine whether a product falls within the scope of GMO regulation or not. So this is an interesting drive towards local innovation, which is also reflected in not only reflected here, but also reflected in the types of organisms that are involved in genome editing in Argentina. So whereas we saw an emphasis on major crops in genetic modification, in genome editing, we see a much more diverse types of organisms involved, including farm animals, including fish, aquatic animals, pasture crops, bioinputs such as nitrogen-fixing bacteria, and so on and so forth. So we see innovation diversity in terms of the actors as well as in terms of the organisms involved. Here's a couple of examples of products that are under development in Argentina. Herbicide-tolerant canola, an important oil crop, is moving forward as a non-GMO because the mutation achieved by genome editing is the same that's already present in the market derived from classical mutagenesis. Argentina is also working on genome-edited tilapia, important fish species for local consumption as well as for export that has improved growth characteristics. And also genome-edited nitrogen-fixing bacteria are approaching the market in Argentina. Again, those products are not developed by foreign multinational, but by local public and public institutions and private companies. Now, these same principles that Argentina adopted are being incorporated as we speak in an increasing number of countries in Africa. I will not dwell on this one too long, but it's really important that not only in South America, countries are convening around this topic and adopting similar approaches to the Argentina regulatory approach, but also in Africa. This is a phenomenon that's occurring in recent years, with an increasing number of countries adopting a case-by-case consultative approach towards genome editing and using different legal mechanisms to do so, ranging from amending an existing biosafety law to issuing guidelines that provide regulatory guidance on genome editing. But in all countries, the regulatory trigger is whether an application or an organism possesses a novel combination of genetic material or not. Now, whereas we all want to stimulate and encourage local innovation and local research, I think I should also give, since I'm based in Europe, also give you an example, quick example from Europe, which has taken a somewhat different approach. And since Europe is an important trading partner for many emerging economies, including the Philippines, of course, it's always important to monitor developments in this, very important part of the world market. Well, as we all know, our history with GMOs is complicated. Only Spain produces a small hectareage of genetically modified maize. It's the only product approved for cultivation in Europe at this point. And this has been grown for the last 25 years, actually and actually quite successfully. Spanish farmers are benefiting from Monet 10 or BT maize to combat the European corn borer. But that's the only cultivation, so we're only a very small player if we compare it to the Philippines. But what we do have in Europe is that we basically contract out GM farming to other countries in the world, because we're heavily dependent on GM commodity imports for our food and feed industry. So if we take the example of soybeans only, the European Union as a trading bloc imports more than 30 million metric tons of soybean products, whole soybeans and soybean meal, of which around 90% comes from countries such as Canada, United States, Brazil, Argentina, that have near 100% GM adoption in their soybean production. So instead of producing our own genetically modified crops, we basically import it from major agricultural exporting countries. So this is important to keep in mind, that more than 100 GM events have already been approved in Europe for importation, for food, feed, and for processing. And that list continues to grow. New events are added almost every month by the European Commission. But one of the key problems that we face here is that our definition of what constitutes a GMO is extremely broad. A GMO in Europe, in Europe's directives, means any organism, with the exception of human beings, in which the genetic material has been altered in a way that does not occur naturally by mating and a natural combination. So we have an extremely broad definition of what constitutes a GMO, which already shows that many applications of genome editing would also fall within this definition. This definition is codified in Directive 2001/18, which is our key biotech directive, which is an example of a very strict process-based regulatory approach. So the product at hand or the characteristics of a product are not assessed, but the method by which it has been produced. I will not go into too much detail as to how we reach decisions in the European Union. I think we need another webinar on that, or a series of webinars. But what's really important is that the scientific advice, the scientific opinions coming from EFSA, the European Food Safety Authority, is just one element in a much broader political decision process, in which all 27 member states and the Council of Ministers and the European Commission, as well as specialized committees composed of member state representatives, have a say on whether a product can be allowed on the European market or not. Well, suffice it to say that a majority vote is never reached. It always ends up in a stalemate, which is why the European Commission, which is the executive body in the European Union, always has to arrive at a final decision, which usually means an approval for imports, but only after very long delays and long and protracted decision-making processes. Some of our applications, some dossiers take 4 to 5 years from the initial application to the approval. And this is just approval for importation and processing as food and feed, not for cultivation. I think it also shows the complexity of our political system here. If you would like to know more about how GMOs are authorized in the European Union, there's an excellent website here that shows you the detailed overview. On cultivation approvals, it's nearly, well, it's impossible to reach a majority among the European Union member states, which is why no company or other applicant even wants to try it these days. So if we look at genome editing or new genomic techniques, as they're labeled in Europe, the debate started well over 15 years ago, because it was clear from the outset that the genome edited products pose a unique challenge to our process-based regulatory framework, because many of the resulting products cannot be distinguished from products derived from conventional plant breeding or conventional mutagenesis. So already in the early 2000s or before 2010, lots of reports were written, meetings were held, and scientific advice was provided on new genomic techniques, after which the Commission referred the decision to the European Court of Justice, who came up with an interpretation of our Directive 2001/18 and concluded that organisms obtained through mutagenesis should actually be considered as GMOs, and that organisms obtained with modern forms of mutagenesis, such as genome editing, are not exempted from our biotech and biosafety directives, from our biosafety legislation. And only organisms that are obtained by techniques of mutagenesis which have conventionally been used and have a long safety record, so that means chemical mutagenesis or irradiation breeding, fall under the exemptions of the directive. So if we go back in our minds to the very broad definition that we have of a GMO here, this is a very logical but very legal interpretation of our biosafety and GMO decision-making framework. And indeed, if we look at the definition, which basically covers all products that are derived with techniques that are not considered natural mating or natural breeding, this is a logical conclusion from a legal point of view. But of course, it presents major challenges to the system, the regulatory system that we have here. First of all, every scientist was up in arms, because what we essentially say is that plants derived from classical mutagenesis, where random mutations are made in a plant's genome, are considered safe. Conventional crosses, which is also a very imprecise technique in a way, are also considered safe in Europe. But if we make one very precise mutation in a plant that doesn't even involve a novel combination of genetic material, all of a sudden we say it's risky and should be regulated. So from a scientific point of view, this process-based approach does not make sense. And more importantly, since detection and labeling is such an important cornerstone of the European biosafety framework, it was also quickly concluded that a big bottleneck or time bomb under our system is that detection of products created by genome editing can often not be distinguished at the molecular level from conventional products or from products that occur in nature. So this is really becoming another very important technical challenge, which is why the Commission, after this initial ruling by the Court of Justice, continued researching and analyzing the situation. Studies were again commissioned on new genomic techniques and the legal implications for the European Union, which was also encouraged actually by the Brexit, the fact that the UK left the European Union and came up with its own policy towards new genomic techniques and genome editing. So the Commission study already concluded that ensuring that European legislation remains relevant is going to be a challenge, especially if the focus is on the techniques used. So not the product, but the techniques, the process used, rather than the characteristics of the final products and the traits they express. So this was already a very clear statement that Europe needs to move from a process-based approach to GMOs and genome editing towards a more trait-based and product-based approach. So the debate is evolving, very recently, the European Commission came out with a legislative proposal that basically divides the products from new genomic techniques or genome editing into two major categories. So the proposal was already leaked before it was published in June. Finally, it was published in early July, and this is the basic tenet of this of the proposal that we currently look at. And what the Commission is proposing now is that products from genomic technologies, so those resulting from targeted mutagenesis as well as cisgenesis, that are legally falling under our GMO directives as ruled by the Court of Justice, will fall into two different categories, with category one that are called conventional like NGD plants or genome edited plants will be exempted from the requirements of GMO legislation. It will require a verification procedure that actually establishes equivalence of the NGT plant with conventional plants, and detailed criteria are actually included in the proposal. A second category involves all other applications, so that are slightly different from conventional products. Those are still considered GMOs, will have to undergo risk assessment under the GMO directives with a case-by-case authorization procedure. Now, this category 2 genome edited products could undergo a GMO light assessment if the changes made in the genome could occur in nature, or if the changes made in the genome pose little to no risk as determined in the initial stages. But all this still remains to be elaborated and discussed, of course, in the various European bodies, as well as in the European member states. What's also interesting here is that plants derived with novel genomic techniques, or NGTs, from both categories are not allowed in organic farming. So this is already ruled out from the outset. Our labeling regime for those products will also be slightly different, where plants falling into category 1, so those that are equivalent to conventional crops, only the seed will have to be labeled as derived from the novel breeding techniques category 1, whereas all the rest will have to be traced and labeled as GMOs. It still leaves open the question as to how traceability can be established, as many of those products will be almost impossible to distinguish from products occurring in nature or products derived from classical mutagenesis or conventional plant breeding. So this gives you an example of how we can stifle innovation by coming up with extremely complicated and process-focused regulations towards genome editing, or novel genomic techniques, as they are called here. So, trying to wrap up here of what I've just said, we've pointed to the very rapidly evolving global context, not just in terms of research and development and product development, but also in terms of regulatory policy. Over the last decade or so, we see a great number of emerging economies, as well as more advanced economies, developing and adopting regulatory policies regarding genome editing. We also look at a very wide diversity of innovators, of trades involved, plants, animals, microorganisms, and this is the type of innovation and diversity in innovation that we would like to encourage as innovators. It is important that countries and perhaps regions, sub-regions, develop their own policies and regulatory approaches fitting with their national goals and priorities, because at the global level, there is no unified approach yet that we can easily adapt to our national situations. A major recommendation from what we see right now is that a case-by-case consultation approach, such as adopted in South America, such as adopted in Sub-Saharan Africa, to determine whether a particular product falls inside or outside GMO legislation is really a very useful approach and commendable approach if a clear set of criteria is developed to make that determination. So governments should put forward a defined set of information and clear timelines in which a determination of non-GMO or GMO can be made. What I find interesting, what we've also just seen, is that approaches in Asia, such as the Philippines, but also in Africa and South America, are aligning much more closely with other emerging economies, such as Argentina, Brazil, as well as others, rather than those being debated in the European Union or under the Cartagena Protocol for Biosafety. What is important, in my view, is to encourage South-South collaboration in this area, as well as sub-regional harmonization through bodies such as ASEAN, such as COMESA and ECOWAS in Sub-Saharan Africa, and also what we've seen in Latin America under their agricultural collaboration agreement. An increasing number of governments in South America are adopting the same principles as Argentina and Brazil have incorporated. At the national level, I think it's important to continue the type of consultations that we have today and encourage coordination among government agencies, as well as capacity development at the regulatory and scientific levels in country. So finally, I think for regulators, we can easily wrap up by saying some criteria that ought to be used in developing regulatory approaches is just, asking ourselves a couple of important questions: whether the product or application at hand contains foreign DNA or a novel combination of genetic material, and if there's a transgene found in the final product or not. That really determines the regulatory approach, and not so much the specific tool or technology used to develop it. Can the edit or the final product also be found in nature through spontaneous mutations? And if so, does it still require a full GMO biosafety assessment? Could the edit or the final product being produced through conventional breeding methods, including mutagenesis, chemical mutagenesis, irradiation breeding? We have a long track record of safety, also with those more crude and far less precise technologies. So if a genome edited product has the same characteristics, should we still regulate it as a GMO then? And a final question is whether a DNA template was used in the repair as a repair mechanism, or is the application based on a natural repair process? So those are important questions that regulators can use in determining case by case whether an application or a product constitutes a GMO or not. So with that, I would like to conclude for now. You have my email address here, and I will be staying with you for, I hope, the rest of the webinar to engage in any discussions or to answer questions. So thank you very much, and over to you, Enrique.
Thank you very much, Sir John, for that detailed and very informative presentation. You presented us from the development in the plant-breeding technology up to EU regulations of or the use of gene editing. So we will now open the floor for questions to our participants. You may raise your virtual hand and ask your question directly to Sir John, or but please kindly introduce yourself. your affiliation before raising your question. Okay. Or you may also type in your questions in the Q&A tab in the Zoom or in the comment section in the NCBP YouTube page. So do we have questions? So sir, Sir comment, Dr. Sir Jan. So you have presented in your slides that PBS provides technical expertise and hands-on training. I just want to ask, maybe it's also some question of some of our participants. Do you provide funding for R&D on agricultural biotechnology research and development in the Philippines or in other countries?
So the question is whether we provide funding or support to research agriculture biotechnology. I think the simple answer is no, because our mandate, our focus is very much on regulatory capacity development. So our products, our final products are much more focused on developing laws, regulations, guidelines, or other policy documents regarding agricultural biotechnology. But I should add right away that especially in sub-Saharan Africa, where capacity in the scientific community as well as in the regulatory community is much less extensive and deep as in a country like the Philippines, also simply because the experience with the technology and with the regulatory questions is much more recent. We do work closely with a range of technology developers, as I would call them, in ensuring that the products from their research are actually entering field trials, multi-location trials, variety registration trials, and so on and so forth, and finally reaching the farmers as new varieties and new brands of seed. So we do support technology development, but from a regulatory perspective and from an outreach communication perspective, I would say. But directly funding research programs is not part of our mandate, but there are other organizations doing so.
Another question, sir. What do you think the possible reason why few multinationals companies are use new breeding techniques or gene editing technology in Argentina? In your presentation, you showed us a pie chart showing. Yep. So why do you think few national companies use gene editing technology compared to GM crops, which is GM? Since it is less rigid if you're going to use the gene editing technology for the regulation.
This is a slide. Oh,
yes, that's.
Sorry, I have to flip back now. Yes, so so if you look at GM crop approvals in Argentina, I think because of the regulatory barriers that we experience with GMOs all over the world, including Argentina, it was only a small number of multinationals who have the resources, the capacity, the data, as well as the financial resources to develop the full dossiers and submit them in different important economies. So the GMO scene was very much dominated by large multinational companies in Argentina as well as elsewhere. if we compare that to the current situation regarding new breeding technologies and products from new breeding technologies in Argentina, we see that well, almost no over 90% of the products under development come from local companies, public research, as well as small and medium-sized enterprises from outside the country that often collaborate actually with local organizations. I should also add that this is not very different from the early days of genetic modification, where we had many small and specialized startups in Europe as well as in the US and elsewhere that experimented with genetic modification technology, and that were subsequently absorbed and taken over by the large multinational firms, again driven by regulatory complexities and regulatory barriers that meant that only the large fishes were able to survive and were able to apply for approvals in major economies. Now, the fact that the regulatory thresholds for genome editing are kept fairly low in a country like Argentina, I mean, within 2 months, you can get an official determination from government whether your technology constitutes a GMO or not. And in many cases, they are exempted from full GMO legislation and regulation, means that it becomes much more attractive for smaller actors to enter the scene and try to develop a fully finished product. Especially in the animal sector, dairy cattle, fish, but also specialty products such as nitrogen-fixing microorganisms. You see a dominance of small, innovative startups rather than large companies. So this is an important difference in the types of innovations we're looking at in this case. This is not unique to Argentina. We see the same phenomenon in the United States as well as in Japan right now.
Thank you, Sir Comment. So last question, we have a question here from NCB people. Gene editing, let me read the question. Gene editing and engineering have applications also in agricultural insect pests. Recently, specifically in South America, there has been a return in the use of sterile insect technique, with the sterility achieved via radiation mutagenesis. What's your opinion on this? Will this affect prospects for gene editing or engineering?
Yeah, of course, if we look at human health applications of gene editing and specifically gene drives, well, I will not be qualified to express an opinion on whether this is a good or bad technology. It's still somewhat controversial, but I do feel it's something we will have to consider and have to test, because of the potential it would have to combat major diseases that right now are an important brake on economic and human development in many countries. Diseases such as dengue, malaria, you name it, are which are transmitted by mosquitoes, are very important economic impediment to development in African countries, for example. So the potential is definitely there. The technology is also evolving very rapidly, from relatively crude applications and techniques such as male sterile technologies, so the release of sterile insect populations, to current experimentation with gene drives that would mean a much longer lasting decrease in disease incidence and have a long-lasting impact on insect populations, impairing their ability to spread diseases. So the potential is definitely there. The technologies are becoming more and more precise, and we're also learning a lot about how, what the environmental impacts will be of, changing entire insect populations. I also feel that for policymakers, this is an important application of new technology, of advanced biotechnology and genetic technologies that may change their thinking about biotechnology and genome editing, because it has so much potential for improving human health. So I'm still hopeful that also here in Europe, the population and the policymakers will start to think differently about GMOs and about genome editing as a result, because it now holds promise for improving human health as well.
Thank you, sir. Thank you, Mr. Comment. So before we let you go, we would like to present this certificate of appreciation for sharing your knowledge on global developments in genome editing applications and regulations, implications for emerging economies during the webinar series titled Biosafety Regulations on Gene Editing Technology. Thank you so much, Sir John.
Thank you so much. It's a great pleasure. I will definitely stay on for a little while longer. I will frame this certificate. Obviously, it's a very important one to me. Next time, I hope to be with you in person, with all of you, and renew acquaintances with many people who are in the room today, as well as meet so many of the extremely talented new generation in the Philippines. So thank you once again for the invitation. It's a great pleasure.
Thank you, sir. So now we will move on to the next presentation on the current status of genome editing R&D in the Philippines, which will be presented by Dr. Reynante Ordonio. So I am honored to introduce to you Dr. Ordonio. Dr. Ordonio is a senior science research specialist and scientist one at the Crop Biotechnology Center in Philippine Rice Research Institute, or PhilRice. He earned his MS and PhD in agricultural sciences at Nagoya University in Nagoya, Japan, where he also conducted his postgraduate program as a visiting foreign research fellow. He is one of the country's leading young rice scientists and was distinguished as the 2021 Outstanding Young Scientist in the field of agricultural science by the National Academy of Science and Technology. Let us welcome Dr. Ordonio. Good morning, sir. Good afternoon. Good morning.
Good afternoon. I hope you can hear me well. So I'm pretty much under the weather right now. I came back homesick from the UK, from my business trip. So please bear with me in this presentation because I'm having difficulty speaking. I might cough in some instances, so please give some allowance. Okay, I hope you can see my presentation now.
Yes.
Enrique
Yes,
is
sir.
sharing it. So let us move on. My presentation is entitled Current Status of Genome Editing Research and Development in the Philippines. So let's just have a briefer about genome editing. Okay, next slide, please. Okay, genome editing is the science of making precise and targeted mutations, such as deletions, insertions, or base changes in the DNA. And this means the DNA that is already present in the genome. We don't necessarily need an exogenous DNA, unlike in the case of genetic modification. This can be achieved through site-directed nucleases, such as meganucleases, zinc finger nucleases, talons, and CRISPR-Cas system. And these SDNs typically have DNA cutting enzyme site we call nuclease that is directed to cleave the DNA at a predetermined location as guided by a specific DNA binding component, producing a double-strand break. And among the site-directed nucleases mentioned here, CRISPR-Cas9 is the most popular. And here we have a short movie showing how CRISPR-Cas9 is cutting the DNA, and it's just a matter of about 30 seconds if you will observe. There's a cut. So next slide, please. Okay, CRISPR is actually already here decades before. In fact, in 1987, this was the first discovery of CRISPR in E. coli. And then in 1993, the discovery of CRISPR. Um, and let's go to early 2000. Sorry, I couldn't read very clearly now. Discovery of adaptive immunity role of CRISPR was in year 2000. And then in 2012, CRISPR-Cas9 hijacked as genome editing tool. So this is the first time that CRISPR-Cas9 was actually used for genome editing deliberately. And then in 2015, um, CRISPR was named as the breakthrough of the year by science. And the history goes on. It's now a very powerful and popular tool for the modern day breeder. So let's move on, next slide. And actually, gene editing, aside from being one of the site-directed nucleases, is also under the umbrella of plant breeding innovation tools. And among the plant breeding innovation tools, CRISPR-Cas9 is the one that is very popular, as we can see here in this trend, which only shows from 2010 up to 2016, but now it's got even more popular. So next slide, please. And the site-directed nucleases produces a double-strand break in the genome, and this is being repaired. And the process this is repaired will dictate how the product will become, whether it is a non-GMO or a GMO. And there are three scenarios involved here: SDN1, in which there is no repair template used, and then SDN2 and SDN3. For SDN1, it is automatically producing a non-GMO. Then in the case in our country, in the Philippines, in our regulation, SDN2 will produce a non-GMO, provided that the insert is just 19 base pairs and below. And for SDN3, this is case by case, and the insertion is greater or longer. And also, we have to take note that since we are using agrobacterium-mediated transformation, the initial stage of product development would involve a GMO. And in later stages, there should be a need to breed out any vector sequences from the product. Okay, next slide. Now let's move on to the different gene editing initiatives in the Philippines. In 2017, ERI was able to publish this paper, and this just cements their pioneering work when it comes to gene editing here in the country. In this particular slide, they're showing rice having lesser number of stomata in the leaves. And this is part of the C4 rice project, in which they're trying to develop rice that would require less amount of water and also less amount of nutrients. So this is a great milestone and also a proof of concept that they can use this in creating C4 rice. And hopefully they will be able to realize that in the near future. Okay, next. Another publication from the International Rice Research Institute is this one from 2018 and entitled Novel Alleles of Rice, EI4G generated by CRISPR-Cas9 targeted mutagenesis confer resistance to rice tungro spherical virus. And through this, they were able to develop lines that exhibits resistance against RTSB. Okay, let's proceed. The next slide. And again, this is another publication from Erie, and this time this is about broad spectrum resistance to bacterial blight in rice using genome editing, in which they edited 3 sweet genes. And this actually means sugar will eventually be exported transporter. That's the meaning of sweet. And by editing 3 different genes, they are creating a broad spectrum resistance in the rice. So next, please click. And this is illustrated in this figure. Kindly click. And there, next, there. So there is a very minimal lesion progression in the tips of the rice in the mutant CS1A, even if different strains of Xanthomonas oryzae were used. So this indicates broad spectrum resistance by editing the different sweet genes in rice. Next slide. And also there are researches in the pipeline now at Erie. One is the BLB resistant gene edited rice. This is actually a continuation of the earlier work I just mentioned. And this is now the application in which they are creating rice in the background of popular high yielding varieties that can possibly be deployed. Well, actually, we know that the first gene edited product to receive the certificate of non-coverage from the ABPI is the gene-edited banana, right? But before that, we were actually eyeing this one as the first one, but it just so happened that there were some issues at Erie that resulted in a delay. But hopefully this will be released or will be applied also for such permit later on and will be released soon. And another is this gene-edited high-yielding rice, which is still unpublished. This is a deliberate loss of function mutation in 1,000 grain weight 6 or TGW6 and grain number GN1A genes using CRISPR-Cas12a, which resulted in increased grain weight and grain number using IR64 variety. So this is also another exciting product that hopefully will be launched by Erie. Next slide, please. And now let's move on to the Philippine Rice Research Institute, particularly the Crop Biotech Center, where I belong. Through the support of the DA Biotech Program Office, we were able to start gene editing work at PhilRice for the first time. And we are now currently working on different traits, such as tungro resistance. This concerns the EIF4G gene. And then bacterial leaf blight resistance concerning OS Suite 14 and OS Suite 13. And when it comes to grain quality, we're also targeting low amylose content in rice with hard texture. And also in response to drought, we are developing deeper rooting rice concerning the OS WEG1 gene. And this is also another one, aromatic rice, concerning the BAD-H2 gene. So these are the different gene editing initiatives at PhilRice, and I'll give you some details in the next slide. Next slide, please. Okay, so these are our putatively edited lines at PhilRice right now concerning the Tungro resistant trait. And the gene that was modified here is the EIF4G. And at the left, you will see the rice that is ordinary rice only and was not inoculated with Tungro virus. And next to it is the wild type rice inoculated with the virus. And so it suffered. It showed some symptoms like stunting and also yellowing. While the rest of the remaining plants here are our putatively edited rice, which are relatively similar to the control rice, even if they were already inoculated with virus. So these again are our candidate lines and will be further tested. And next slide. This now are our putatively edited lines involving the editing of always sweet 14. On the left is the control rice showing a high degree of progression of the lesion after inoculation with Xanthomonas oryzae, and next to it is the control in which it is scored actually as resistant because of the minimal progression. And the three remaining ones show this one is the middle. At the middle is the original rice before editing, which shows a high degree of lesion. And then next to it is the moderately resistant putatively edited line. And lastly, our putatively edited line with resistance. So that's how we are generating the lines and to be further tested. Okay, next slide, please. Now let's move on to the Institute of Plant Breeding at the University of the Philippines in Los Baños, in which they are currently developing non-browning eggplant. And right now, they already have T1, up to T1 plants, which are being forwarded to T2 generation. And from there, they would be doing assay to really determine and to screen for plants for fruits that show non-browning, and then they will forward them. Okay, this is very exciting too. And aside from that, of course, we know that they are developing the BT talong, which is a GMO. Okay, next slide, please. And also from the IPB, they're developing low phytic acid corn. Phytic acid is an anti-nutrient, so it's important to reduce it in corn. And they're doing that by targeting corn IPK1 gene to reduce the phytic acid content. And then the high lycopene tomato, they are aiming to increase the lycopene content by knocking out the gene that is involved in the accumulation of or to the accumulation of beta carotene. Instead of accumulating beta carotene, it would be transformed into lycopene, which results in redder tomatoes. Next slide, please. Okay, so from here, we can recognize that the three institutions that are mainly involved in gene editing or actually doing gene editing in the Philippines are ERI, PhilRice, and IPB, or the University of the Philippines. And we can have some insight on why they are the ones that are doing gene editing from these succeeding slides that I will show you. These are the different technical considerations that must be considered or met in doing gene editing. Number one is that full genome sequence of the target crop must be available. Luckily, in rice, this is not a problem because the genome of rice has already been elucidated long before. And it's not so surprising why rice is the first one to be edited in the Philippines. Number two, availability of functional tissue culture protocol for the crop for regeneration. So for the crop, there must be this protocol. And even in rice, actually, even if it has been being edited for some time, there's still a problem doing regeneration using some indica rice varieties. So this is still open for optimization. And the number 3 target gene must be well characterized in terms of biochemistry, molecular biology, and genetic, and the way it translates into the expected phenotype must also be well characterized. And then licensed gene editing vectors must also be available in order to do transformation and gene editing, because this may affect product commercialization later on, depending on your on the license that is given to you. And next slide, please. Okay, these are other considerations as well. There must be ample funding. Number 2, highly skilled and trained personnel with relevant technical expertise, such as in genetic transformation. Number 3, knowledge of GM regulation also, because GMO are being generated during the early stage of uh, product development in gene editing. So this is also another important knowledge that must be in the technical persons that are doing the gene editing and the team also. And the number 4, approved biosafety facilities. So there must be an accredited BL2 laboratory and accredited greenhouse. So from here, we can recognize that the 3 institutions all have prior experience when it comes to like GMO regulation, and they have invested on considerable number of biosafety facilities, and they have trained personnel, and that's probably the reason why it's very easy for them to move on to the next level, which is gene editing. And this is very fortunate also for Phil Rice, because we have been working with Erie on golden rice and also high iron in zinc rice, and with ample funding coming from DA Biotech, we can easily move on to gene editing, doing gene editing. And also especially that there is the Crop Biotech Center right now through the help of US funding and also by the government. The number 5, basic laboratory equipment for tissue culture, DNA processing and analysis, and bacterial culture must also be available. And presence of a functional institutional biosafety committee. This is also very important. And I think this is also one of the setbacks or hurdles for some other institutions to do gene editing as well. Number 7, institutional support commitment from research, varietal release, and beyond. So this is something that Phil Rice is very fortunate of, because the management leadership at Phil Rice is very supportive and committed in helping the researchers really progress with the work. Like in the case of golden rice, uh, there had been a lot of issues with golden rice, and still the management is very supportive. And the number 8, public support and acceptance. This is also another important consideration. And for an institution that is part of the government and is a reputable one, like Phil Rice in Erie, it should be easy to have public acceptance and support. But and most especially if we're talking of gene editing, it is understood that, or understandable that for golden rice there had been a lot of issues, but since we stick with the science and we um, we stand with our reputation and we adhere to integrity, then it's very important, and this led actually to more public acceptance. The number 9, gene editing policy in the country must be in place. This is another very important consideration, and we're very lucky that since 2020 up to 2022, we have set the necessary policy and then the guidelines for allowing the application from companies like Tropic with their gene-edited non-browning banana, which have been approved in just 26 days. This is very important. So this is very lucky for the Philippines is very lucky that we have now this gene editing policy. And then number 10, insurance. I will leave this to your imagination what aspect of the research must be insured. Okay, next. So here are some key takeaways. Genome editing of crops is being carried out by 3 institutions in PH, particularly Erie, DA Pilrice, particularly DACBC, and UPLB, including the Institute of Plant Breeding, with Erie being the pioneer. Most of the researches involve SDN1. So this is the basic gene editing in which there is no introgression of the repair template or exogenous DNA in the final product. And then rice is the main target crop in the Philippines. And lastly, some advanced lines are already nearing varietal registration, like the BLB resistant rice that is being developed by Erie, and hopefully the Tungro resistant and the BLB resistant rice we're also developing at Pale rice, and of course the gene edited non-browning eggplant from the University of the Philippines. So with this, thank you so much for your attention and for your listening.
Thank you so much, Dr. Ray, for the presentation. Yes, you mentioned that gene editing policy is already in place in the country. And this policy or resolution and plant breeding innovations or new dairy techniques will be tackled by our next speaker. So for now, we will entertain questions to our participants. You can raise your virtual hand, or you may key or type in your questions in the Q&A tab in Zoom or in YouTube comment section in NCBP YouTube page. So I'm just curious about the gene edited high yielding rice and gene edited or non-browning eggplant. Are they considered GMO or non-GMO?
Come again. So you're probably asking if the non-browning eggplant and high-yielding rice are non-GM. I also checked, and they are non-GM because they are involving SDN1 of gene editing. There's no exogenous DNA being introduced in the final product.
Sir, Dr. Ray, I have a question. So, good morning, everyone. I'm Raymond Corona from DOST-NCBP. So, I'm asking this question on behalf of the state universities and colleges here that are in the attendance, that are attending our webinar. So, is the Crop Biotechnology Center currently have programs for capacity building for state universities and colleges on basic, intermediate, and advanced courses on molecular biology with focus on agricultural crops?
Yeah, when it comes to molecular biology, CBC has actually been accepting trainees from universities. We had universities from Pampanga, from VSU, from the Visayas State University, and already about 3 or 4 batches have been completed. So we're also still very much accepting of trainees for as long as it will not affect our regular work. We will schedule them.
All right, that's good to hear, sir, because even in our last webinar 2 weeks ago, that's one of the most prevalent questions from researchers from state universities and colleges. And it's a good thing that Craft Biotech Center is accepting trainees for the specific purpose.
They are the OJTs.
All right, sir. Thank you very much. That's very nice to hear.
Hello? Yeah.
So now we proceed to our definitely not the least the first speaker.
We can't hear you.
Hello. So now let's proceed to our next last speaker, the first speaker. She is Ms. Darlene Joy Sena. Ms. Darlene is a science research specialist, one of the Bureau of Plant Industry Biotechnology Office of the Department of Agriculture. She conducts compliance monitoring activities for approved genetically modified events, site monitoring, bioefficacy studies, and other biosafety activities. She also reviews and creates technical materials for the Biotechnology Office and the National Urban and Peri-Urban Agriculture Program. Ms. Darlene is also a registered agriculturist and a fifth placer during the licensure examination for agriculturists in 2018. Ladies and gentlemen, please welcome Ms. Darlene Joy Sena with her talk on Philippine policy for products of plant breeding innovations, or PBS. Ms. Darlene, the
Yes, sir,
virtual.
thank you. Thank you so
Hello.
much for the introduction. So let me share my screen. Okay. Can anybody tell me if you can see the presentation?
Yes, we can see it, Ms. Darlene.
Okay, so thank you, Sir IQ. So good afternoon, everyone. So again, I'm Darlene Joy Shana from the Biotech Office, and this presentation was actually initially arranged to be presented by the head of our office. However, not unfortunately, but she has to attend to an equally important matter, and I'd like to thank Ma'am Emma and the organizers of this webinar for entrusting me to present this topic to you this afternoon. So we'll be discussing the Philippine policy for products of plant breeding innovations. So if you have been with the NCBP webinar, if you have witnessed the previous part last 2 weeks ago, and even as discussed by Dr. Ray Ordonio earlier, there are several techniques that is being applied for plant products to be modified on the genetic level. And this just shows that biotechnology is really an evolving field of science, and the regulation and policy that governs it really needs to be updated and should be aligned on the science behind it, or the changes that is happening with it. And luckily here in the Philippines, we have this policy to cover or to govern these PBI products. So just to give a brief background on how this was developed, way back in 2018, the Department of Agriculture commissioned a study together with esteemed scientists and regulators titled A Review on the Plant Breeding Techniques from the Viewpoint of Regulation. So the main objective was to check whether the PBI product would contain novel combination of genetic materials, or what we call as transgenes, or not. The result of the study was then submitted to the NCBP, or the National Committee on Biosafety of the Philippines, and they used this as a basis for the issuance of the NCBP Resolution Number 1 Series of 2020. So I am showing here a part of the resolution, or the signature page, and 6 secretaries from different departments were signatories, as well as 1 undersecretary, and then other members of the committees were also there to sign this resolution. And it has 3 major points. First is that it resolved the classification of PBI products. So according to the resolution, there are 2. First is that either it will be GMO or non-GM. Simply put, PBI products with transgene or novel combination would be classified as GMO. If they don't have transgenes or novel genetic composition, it will be considered as non-GM. Then the second major point is that PBI-derived GM plant products or plants would be under the scope of the JDC number 1 series of 2021, which is the regulation for GMOs here in the Philippines. If the PBI-derived product is non-GM, it will be considered as a conventional product. So JDC won't apply to them. And lastly, or the third point, is that the DA shall issue guidelines and take the lead in evaluating and monitoring plant and plant products derived from the use of modern biotechnology, including PBIs or NBTs. Okay. So moving forward, after 2 years of the issuance of the resolution number 1, the Department of Agriculture now issues the Memorandum Circular number 8, series of 2022. This took effect last year around May, if I recall it right. And this is titled as Rules and Procedure to Evaluate and Determine when Products of Plant Breeding Innovation or PBIs are covered under the DOST, DNR, DOH, DILG Joint Department Circular number 1, series of 2021, based on the NCBP Resolution number 1, series of 2020. So simply put, this serves as a guideline for both regulators and applicants on what the procedures are or the requirements are for us to conduct a technical determination on the PBI products regulatory status. So it will guide us on determining whether they are GM or not GM. So this will be the whole coverage of our slides later on. So just a short definition of what PBIs are. So this has been discussed by earlier speakers more thoroughly. So PBIs are defined as a set of new molecular tools that enable targeted and efficient development of new varieties of crops with desired traits in a way that is faster and more precise than conventional plant breeding techniques. And of course, one of the most prominent or most used is the CRISPR. And this has been also explained to us by our resource speakers before. And to reiterate, same with the resolution number 1 series of 2020 from the NCBP, the DAMC number 8 series of 2022 also follows the definition set by the earlier resolution. So it says that there are 2 general classifications of products of PBI. It can be either GMO if it has transgenes or novel combination of genetic materials. And if it does, it will fall under the JDC. And if it is, if the PBI product doesn't have or don't have that novel combination or don't have the transgene, it will not fall under the scope of the JDC. It will be treated as a conventional product. Okay, so now let's go into the overview of the processing of submissions. So the Memorandum Circular number 8 calls for about 27 to 32 working days of total processing time. After that, you have to have a official determination already of the regulatory status of the product. So we have 4 main stages. First is the pre-assessment. So that takes around 3 working days. So this happens in the biotechnology office here at the Bureau of Plant Industry. The product developer would submit the required documents and the biotech office would check for the sufficiency in substance and form of those documents. And if they see that it is complete and suffice in form and substance, the BPI shall issue an acceptance letter to the product developer indicating that we are going to start processing your request for the conduct of technical consultation of evaluation and determination. So it's quite long, so we just call it as the conduct of dissent. So after that, the office has 3 working days to forward the documents to the evaluators or those that will do the dissent. So the dissent or the conduct of dissent will take about 24 working days. And simultaneously happening with that is the public commenting period. The result of the TICED or the recommendation from the TICED group, along with the result of the public commenting period, will then be forwarded to the BPI director for him to use as basis for the official determination that will take for around 5 working days. After that, he has to decide whether the PBI product is a non-GM or a GM. So who may submit requests for TICED? So product developers who intends to introduce a PBI product into the country shall submit a request to the director of the BPI for technical consultation for evaluation and determination. So there are 2 classifications. Those that are residents or these listed groups or institution are considered to be natural or juridical person may submit. However, for companies that are coming outside the countries, what they can do is to appoint an agent who is a resident of the Philippines and who will be in charge of the submission or the processing of the TICED. So listed here are the requirements that a product developer must submit. First is the TICED form, and then the prior evaluation form and supporting scientific studies. So again, if the product developer finds that the submission is lacking in form and substance, the submission will not be accepted. So we have 3 working days upon the acceptance to forward this to the BPI Biotechnology Core Team, Plant Breeding Innovation Group. This is the sample of the form. So we have here the TICED form. So they have to answer the details of the product developer and the agent, if it's applicable, then a short description of what the PBI product is, the phenotypic feature before and after the genetic modification or change, so they will be explaining that as well. This will be signed and notarized by the product developer. And then this is the next form that they have to submit, the prior evaluation form or PEF. So this is more detailed on the genetic modification and the other evidences. So as you can see here, there are description of PBI product that is being required, and then the description of PBI procedure that is used or to be used. And yeah, the guide RNAs, the nucleases, the delivery system. So these are very highly technical and on the molecular level aspect of the PBI techniques. They also have to give us the evidences or experimental evidences showing that the final product do not contain transgenes or novel combination of genes. So they have to expound it here and show us proof. And then of course they have to list the scientific studies and other evidences here. So this is the portion for the BCT-PBI TICED group where they will place what is the recommendation or what is their determination for that specific PBI product. So if they will check if this is a GMO, they will check it here, and if it's not, the lower box. They will print their names and signatures, and then this form will be forwarded to our director as basis for his final determination or official determination. So who will conduct the technical consultation of evaluation and determination, or the TICED? So here in the BPI, there is a group that we call as Biotechnology Core Team. These are composed of several personnels from different divisions of the Bureau of Plant Industry. They have backgrounds in biotechnology, on insect management, on weed management, and other related sciences that is necessary in the implementation of JDC and this new circular. So within that group is a specific group that focuses on the implementation of this DAMC number 8 series of 2022. And we call them BCT-PBI group. Okay, so for each application, for each submission rather, it is required that the BCT-PBI will form a TICED group. that will be the one to conduct the technical determination of that certain submission. And the members of the TISED is set to be composed of 3 members. At least 2 should come from the BCT PBI. If needed, they can add an external expert with backgrounds on molecular biology, bioinformatics, and other sciences that would help in the conduct of the TISED. So we have 3 for the TISED group. And then during the conduct of the TISED meetings will be scheduled by the BCT PBI chair. So that will take around 24 working days. And then after that, they should have come to a conclusion on what their determination is for the BPI director. During those meetings, since our timeline is very short, we have to involve the product developers. So the product developers are mostly invited during the meetings. They can come through face-to-face or through online conferences for the TISED group to immediately raise questions or request additional information if it is needed for the conduct of TISED. So the product developer is given 5 working days for him to give the requested additional information. This one is the decision 3 that is also used as reference for the reference of the TISED group. So actually, the techniques that you have encountered or discussed by our previous speakers are listed here, and it shows you what is the expected resulting product, whether it is GM or non-GM. While this is used as reference, is still during the conduct of the TISED, what is being considered is the final product. What is the genetic composition of the final product rather than the process itself. So here in the Philippines, compared with what Dr. Komen has discussed earlier for EU, we are focused on the final product of the PBI when conducting the technical determination. And then we also have the public commenting period. So accepted submissions will be posted immediately on the BPI website, and the public may submit to the BPI director any technical information on the submission within 10 working days upon posting. So this technical information from the public will also be used as basis of the director for his official determination. So we will be forwarding the recommendation from the TISED group and the simultaneously conducted public commenting period to the BPI director, and then he is given 5 working days to give his official determination. If the BPI director determines that the PBI product is a GMO, the BPI will inform the product developer in writing that the GM PBI product is under the scope of JDC, and we will advise him to go through that process. BPI will also monitor this one. And if it is found that the PBI product is non-GM or conventional product, the PBI product will be given the certificate of non-coverage from JDC number 1 series of 2021. This certificate will be made public through posting in the BPI website for transparency. But the certificate will not excuse the product developer from complying with other relevant regulations that he needs to comply here in the Philippines. The memorandum circular number 8 series of 2022 also has a clause for appeal. So any aggrieved party may file an appeal with the DA secretary 15 days from the receipt of the product developer of the decision or the posting of BPI website of the certificate of non-coverage. So that's all from my end. If you want to contact our office for questions or clarification, you can reach us through this email address, and you can also check our website for more information regarding regulations of GM and PBI products here in the Philippines. So thank you very much for your attention, everyone.
Thank you so much, Dardine, for that thorough discussion on the process of the applications for PBIs. So we will now open the floor for questions. Once again, you may type in your questions in the Q&A tab or comment section in the NCBP YouTube page. Okay. So, do you have questions?
Well, ask the first question. So thank you, Darlene, for the presentation. I guess just to give more, how do you call this, appreciation on the process of the efficiency of the DAMC circular number 8 series of 2022. Can you give us some anecdotes on your experiences towards the determination of land coverage for the tropic biosciences banana?
Okay, so what Sir Raymond is talking about is the first application actually of BPI for PBI product determination. So we accepted that we have released the certificate last March 15, if I'm correct, for that product. So since it was also challenging for our end since that was a first application. So what we actually did was to meet up with the product developer initially before they submitted it and requested for what do you call this, preliminary presentation of the procedures that they did for the product. So yeah, that's it. So it was really challenging, but we're really glad that we were able to produce and to really have a determination that is science-based and that follows the M's.
Thank you. So regarding that, how many application or submission that BPI received for BPI product determination?
Yeah, currently we have received one for the reduced browning banana, and we have a determination already for that. Yeah, so currently we are actually we are just fixing some regulations, a policy regarding the fees of for this MC, and we were advised to we're just waiting for that. But I think there are also planned submissions from the end of product developers.
That's question. I'm curious about the public participation. So in the case of the GE banana, is the public really give comments during public commenting period?
In the case of reduced browning, we didn't receive any comments from the public for that. Sir IQ, sorry, are you saying something? I can't hear you properly.
Yeah. Can you hear me now?
Yeah, I can hear you now.
So I'm just reading the comments of Sir comment in the chat box. So okay, only one PBI determination so far po, and a very clear policy and excellent presentation.
Thank you, sir. Thank you po.
Commented. Okay, I think we have no more questions. So we would like to present the certificate of appreciation to Ms. Darlene for sharing her knowledge on Philippine policy for products of plant-breeding innovations, or PBIs, during the webinar series titled Philippine Biosafety Regulation on Gene Editing Technology. Okay. Thank you, Ms. Darlene.
Thank you po. Salamat.
Thank you very much, Mr. Komen, Dr. Ray, and Ms. Darlene for accepting our invitation for this webinar, for sharing your knowledge and experiences in gene editing applications and regulations. And thank you for all the participants for attending this second webinar of NCBP and PBS webinar series. So to officially conclude today's webinar, it is my pleasure to call Mr. Raymond S. Corona, Project Senior Technical Specialist of NCBP, to deliver the closing remarks.
So thank you, Sir IQ. I won't hold anyone much longer. So on behalf of the National Committee on Biosafety of the Philippines, thank you, John. Thank you, Dr. Ray. Thank you, Darlene, for accepting our invitation. Thank you to Carlo, the country coordinator for PBS. They have been our partner for the past 2 decades, and we look forward for 20 more years, 40 more years of engagement with PBS. Thank you to all the participants from the state universities and colleges, public research institutions. You really are our intended audience for these kind of activities. We intend to reach out beyond the confines of Metro Manila and Luzon in terms of capacity building and knowledge sharing and knowledge and mainstreaming of biosafety and modern biotechnology in our country. I would like to reiterate that our office is always open for any inquiries regarding modern biotechnology, biosafety regulations in our country and internationally. You can reach us through our email address, through our contact information indicated in the emails that you have received. And we look forward to your continuous participation in this series of webinars. We'll keep you posted on the next set of topics and speakers that the NCBP would offer probably the month of October until November. So once again, thank you very much.
Thank you very much, Raymond, for that wonderful message. So before we end today's webinar, we would like to request everyone to turn on their cameras or video for photo opportunities. Sino yung camera? Okay, panel 1, smile. Okay, second panel. And last panel. Okay. Okay, thank you. So, thank you, everyone. So we hope you can still yes, for the evaluation, kindly, in order to receive, to all our participants, for you to receive a certificate of appearance or attendance, you can please accomplish first the evaluation form in the link provided in the chat box in the Zoom. Okay. So we hope you can still join us for the upcoming third part of this webinar series. So that concludes our webinar on the Philippine Biosafety Regulation on Gene Editing Technology, brought to you by the National Committee on Biosafety of the Philippines and the Program for Biosafety System. So have a great day, everyone. Bye.